So good afternoon, everyone, and happy World Malaria Day. And it's a real pleasure to be here today and share this day with you, and also share some of the work that we are doing at Harvard on the biology of the mosquitoes that transmit malaria, and also how our studies on the mosquito biology are leading to new ideas for possible new methods for mosquito and malaria control. As we've heard this morning from Hillary and others, mosquito control is central to malaria control strategies, and it's been postulated to have contributed a lot to the decline in malaria cases that we've seen in recent years, with especially the use of bed nets has been a major contributor in the decline of malaria cases since the beginning of this century. However, as we've heard all morning and this afternoon, malaria cases have sort of plateaued. They're not going down in recent years. And one possibility is that this is due to the widespread insecticide resistance in mosquito populations that might be endangering our best control tools. So we don't really know the public health impact of this insecticide resistance in mosquitoes. However, it is possible that it's at least partially contributed to the less efficient control of malaria that we've seen in recent years. But there are also other limitations with the use of insecticide-based strategies, is that both bed nets and indoor residual sprays target populations that feed and rest inside our homes. And so we don't really have good tools to target mosquito populations that feed and rest outdoors. So pretty much everyone will agree that we need new tools for malaria control. And in my lab, we study the basic biology of the interaction of mosquitoes with parasites in both laboratory studies, but also in field settings, mostly in Burkina Faso, to try and identify new concepts, ideas, and also possibly tools. And in recent years, we've been looking in particular at this interaction between mosquito reproduction and parasite development. And the reason for this is because the two processes, the processes of mosquito reproducing and parasite developing in the mosquito are quite linked, at least temporally, because they happen pretty much at the same time. So a mosquito feeds on blood because she needs to reproduce, and she uses the lipids and other resources from the blood meal. Maybe I shouldn't try this. I'll try. Yeah. Anyway, to develop eggs over the course of 2-3 days. So she accumulates lipids and other resources into the developing eggs during the first couple of days after blood feeding. And at the same time, if that blood is infected with malaria parasites, also parasite development will happen in the mosquito with the fertilization of male and female gametes to form oocytes, that then will, as you know, transform into oocysts, which will then sit for about a week underneath the basal lamina of the gut, and then producing hundreds or thousands of sporozoites that then will reach the salivary glands for where they can be injected again. And so the parasite cycle in the mosquito lasts for over 10 days, and this is quite critical for malaria transmission, because most mosquitoes don't live that long, or the mosquitoes don't live that long in the field, and so the speed of parasite development is quite critical for their ability to transmit malaria, and this will become relevant later. But so one thing that you will notice from this graph is that really the earliest stages of parasite development temporarily overlap with the stages of egg development. So one of the questions that we are asking in the lab is whether there is an interaction between these two processes. And these are studies that a few people in the lab, Christine, Rob, Maurice, and Perrine, have been doing in the last couple of years. And so we've been asking whether the development of the parasite is affected by the development of the eggs, and vice versa, whether the development of eggs is affected by the presence of parasites. So whether mosquitoes somehow are affected by whether they are infected or not. And when we do these studies in Anopheles gambiae mosquitoes, which are the most important vector in Africa, and we use Plasmodium falciparum parasites from the same regions, we see that mosquitoes don't really seem to suffer cost of infection. So whether they are infected or uninfected, as you can see here, these are uninfected or infected mosquitoes, they develop pretty much the same number of eggs, which suggests that they might not be really suffering fitness costs. And we can go a step further and take the infected mosquitoes and look at the number of eggs they develop in the ovaries and the number of parasites that they develop in the midgut, and then look at whether the two are somehow correlated positively or negatively. And to our surprise, what we find is that there is a positive correlation between the number of eggs shown in the x-axis that a mosquito develops and the number of oocysts that she can harbor in the gut. So a mosquito that is more fit seems also to support more parasites. And this is in our laboratory studies using cultured NF54 parasites and also mosquitoes that have been in the lab for many years. But it's the same if we go to the field. We do field studies in collaboration with Abdullah Diabaté and Thierry Lefebvre from IRD and IRSS in Bobo Dioulasso in Burkina Faso. And we see the same positive correlations, more eggs, more parasites. Which also means that there are females that have fewer eggs and fewer parasites, which could represent some good news that if we reduced the ability of mosquitoes to produce eggs, we could also negatively impact parasite development. So this is one of the questions that we've been asking. And to do that, we can manipulate mosquito reproductive processes and reduce their ability to develop eggs, and then see what happens to the parasites. And we can do that by multiple means, but for the sake of time, I'll just show you one such strategy that we can use. We can target the function of steroid hormones, particularly the steroid hormone 20-hydroxyecdysone, or 20E, which is a hormone that is produced by the female mosquito after blood feeding, as you can see here, 24 hours roughly, and it's produced by the mosquito fat body. And this hormone is a potent transcriptional regulator and regulates the expression and also the function of lipid transporters that then will mediate the incorporation of lipids into the ovaries, so inducing egg development. So if we interfere with the function of this steroid hormone, we also interfere with egg development. And we can do that by preventing the interaction of this hormone with its nuclear receptor, ECR, by RNA interference. So we can inject females with molecules that interfere with the expression of the ecdysone receptor and then infect these females and see what happens to eggs and parasites. And when we do that, we see a decline in the number of eggs, as predicted, but we also see that on the left-hand corner that those mosquitoes also harbor fewer parasites. So fewer eggs and fewer parasites. And at the moment, we're trying to understand why they develop fewer parasites, what are the mechanisms by which parasites are killed, but this is not the scope of this talk. I will not tell you much about it for the sake of time. But anyway, this sort of was encouraging for us that we could target mosquito reproduction and at the same time also negatively affect parasite development, at least in terms of numbers, because the number of females that are actually positive for parasites, so the prevalence of infection is the same in two groups. However, the mosquitoes in red, where we interfere with egg development, also show fewer parasites. But, however, the situation is a little bit more complicated because when we started looking at those parasites a little bit more closely, we realized that they look different. And in particular, they look bigger. So if we look at oocysts in the midgut of control females on top or females where we manipulate the ability to develop eggs, we see that oocysts look bigger under the microscope. And then we can measure correctly and precisely their size throughout development, as shown on the left. So the oocysts from the 20E signaling impaired females are in red. And so we can see that females in those mosquitoes the parasites in those mosquitoes are always larger across different days after development. Pretty much from day 5 onwards, those oocysts look larger. And so what does that mean? Larger oocysts might mean oocysts that are growing faster, that have a faster developmental rate. And the way to determine this is to look at the next developmental stage, which is the sporozoites. And indeed, what we see is that females here that have reduced egg development show already at an early day, 10 days after infection, show quite a large number of sporozoites in their accessory glands compared to females that develop a normal number of eggs. And also, so the number of sporozoites in those females is larger already at day 10. So there are more females that are infected with sporozoites, and moreover, they have more sporozoites in their accessory, in the salivary gland, which to us suggests that really those sporozoites and those parasites in general are going faster in those mosquitoes. So they reach the salivary gland sooner. And moreover, they not only are faster, but they keep their infectivity to humans. We can do these experiments in collaboration with Sangeeta Bhatia at MIT. So we can take salivary gland sporozoites in different groups of mosquitoes and then use them to infect the primary human hepatocytes that are grown in Sangeeta's lab. And what we see on the left is that the two groups of sporozoites, those that are fast, let's say those that are slower, are as infectious to primary human hepatocytes. So these sporozoites go faster, they grow faster. However, that doesn't occur to the detriment of their infectivity. They are still as infectious. So this is very interesting from a biological point of view, that we can manipulate mosquito reproduction and see an effect on the parasite. But it's also a little bit a concern for if we think of strategies that aim at reducing the ability of mosquitoes to reproduce, such as population suppression gene drives, where we will see a decline in the number of eggs and possibly also a reduction in the number of parasites, but those parasites will be transmitted faster. And so the consequences for malaria transmission could be negative. So it's something to keep in mind when manipulating mosquito physiology, that we might see unintended consequences. And this is also even more relevant because we see exactly the same phenomenon if we do these manipulations in mosquitoes that are field-derived, and if we use parasites from gametocyte carriers, so field infections, to infect those mosquitoes, as shown here on the left. So we can take blood from gametocyte carriers from villages near Bobo Dioulasso and use those infected blood to infect different mosquito species, in this case Anopheles gambiae, and the sibling species Anopheles collucii, so two very related species, and we see exactly the same phenomenon. We see that parasites grow faster in mosquitoes that have fewer eggs. And so this is, again, very interesting, but it's also sort of a challenge in terms of malaria control, because what we see is that when we try to target the mosquito directly and we try to kill mosquitoes, they might develop resistance, for instance, to the action of insecticides. But also other types of manipulation of mosquito physiology might affect the parasite. And so the parasite has the ability to adapt to manipulations in mosquito physiology in ways that are not always predictable and not positive for malaria control efforts. And so these observations made us think that maybe there might be other ways that we can block malaria transmission without really affecting the mosquito vector itself, at least not too much. And so let me take a step back and look at what type of control strategies we're using currently in the field. So for mosquitoes, something happened to the picture, but it looks nice here. So for mosquitoes, we use insecticide-based strategies, whether bed nets or indoor sprays, to kill them. And how these strategies work, mosquitoes land on surfaces that are treated with insecticides, and then they can absorb those insecticides through their legs. Insecticides normally are lipophilic compounds that are taken up by the female mosquito or the male mosquito through the legs, and this kills the mosquito. While in humans, obviously, we use antimalarials that can kill parasites that are in circulation. And so the idea that we had is, like, could we actually merge these two strategies and put antimalarials on mosquito nets or other surfaces, and so that the mosquitoes can take them up through the legs the way they would take up insecticides, and we can maybe kill parasites inside the mosquito rather than killing the mosquito itself. And so this sounded like a bit like a crazy idea, but I managed to persuade one of my postdocs, Doug Payton, to try. And Doug started using a particular antimalarial, atovaquone, which is a cytochrome B inhibitor, is a potent antimalarial, as you know, is used for prophylaxis in travelers. But it's also a highly lipophilic compound. And so Doug, as a proof of principle, thought that we could use atovaquone, and because mosquitoes might be able to take this compound up. And so to look at whether we could kill parasites within the mosquito. And so what Doug did, he coated a glass surface with atovaquone for 60. So I coated a glass surface with atovaquone and placed mosquitoes for 60 minutes, so for an hour, on this glass surface. And then after exposing mosquitoes to atovaquone, he blood fed them on infectious blood. And then after a few days, he went on and looked at the number of oocysts developing in those mosquitoes. And to our surprise and delight, we found no oocysts whatsoever in those mosquitoes. So while in the control, the vast majority of females, almost every female, was infected with malaria parasites and also with quite a few parasites, in the atovaquone-exposed females, we could see no parasites whatsoever. And then Jack decided to reduce the exposure time down to 6 minutes, and he found exactly the same effect. So 0 parasites so none of the mosquito was infected in the atovaquone-treated group. And then we also did a 10-fold dilution series and found that the compounds were still very effective, also at quite low concentrations, and at concentrations that are comparable to the concentrations of insecticides that are placed on bed nets. The same effects he found when, rather than using glass surfaces, he put the antimalarial directly on nets, and again, 100% killing of the parasites. And then he started also playing with conditions. What if we expose mosquitoes a day before, what if we expose mosquitoes a day before infection or a few hours after infection? And again, the same results, so no parasites present in those mosquitoes. So these very, very promising results. But what about the mosquito itself? Is the mosquito affected by taking up this compound? And the answer is no. We can see no effects in terms of mosquito longevity on the left or egg development on the right compared to controls. So there seems to be no fitness cost to the mosquito after taking up these compounds, which is sort of promising, because then the mosquito will not be stimulated to have resistance, to develop resistance to this strategy, because they're not affected whatsoever. And so with these results, we went to our colleagues in the epidemiology department, Caroline Backey and her former postdoc, Lauren Childs, who's now at Virginia Tech, and we asked them to model the possible effects of adding antimalarials on nets. And what they did, they used data from about 500 locations in East and West Africa. And in these locations, we have data on malaria prevalence, we have data on bed net distribution, and we also have data on insecticide resistance. And so they inputted all this data, and then they asked the question of what difference would it make in terms of malaria prevalence if those bed nets also had an antimalarial that can kill parasites. And as you can see, in both West and East Africa, the model predicts that the addition of an antimalarial that can kill parasites will be quite potent in terms of reducing malaria prevalence in those settings, which is something that is very promising in terms of the possible application of a strategy like this in the field. And one thing that we're doing at the moment, because obviously atovaquone is a drug that is currently used for prophylaxis, and so we wouldn't want to endanger its use as a malaria therapeutics. So we are collaborating with different, with MMV and GSK and other partners in trying to screen and select for additional antimalarials that can kill parasites in the mosquito to move this strategy forward. But I don't have data yet to show you, maybe in a year's time. And also, we've been asking whether this strategy could actually work in field settings, and especially in mosquitoes that are highly resistant to insecticides, and with gameticides and with Plasmodium isolates that are currently circulating in Africa. And so again, in collaboration with Abdullah Diabaté and Thierry Lefebvre in Burkina Faso, Bobby de Lasso, we've been performing these experiments by collecting mosquitoes from natural breeding sites, in this case rice paddies near Bobo Dioulasso. Then we can take larvae from those breeding sites, take them to the lab, and expose them to atowacone, and then infect them with blood from gametocide carriers, and then ask the question of whether this is actually something that could also work in insecticide-resistant mosquitoes, and also against circulating Plasmodium falciparum isolates. And we tested these mosquitoes for their resistance to insecticides, and that actually, you cannot kill them. Like, we have to expose them for 5 hours to 5 times the dose of permethrin before we can kill them. So the resistance levels is really, really high. And the results that we got a few months ago show that this strategy is also very, very effective against field parasites in insecticide-resistant mosquitoes. So we got only a few mosquitoes that were infected, and so just a few of those mosquitoes were infected compared to the vast majority of females in the control experiments. And also those that were infected had only like 1 or 2 oocysts in their midgut compared to quite a lot of oocysts in the controls. So this is something that could also potentially work in field settings against circulating parasites and in mosquitoes that are highly resistant to insecticides. And so this is pretty much what I wanted to share with you today. So as I showed you, and as we all know, targeting the mosquito is challenging, and also we see that parasites can adapt and can respond to changes in mosquito metabolism that we might impose with the vector control strategies. And we provide proof of principle that drugs can be used to kill parasites directly in the mosquito without affecting the vector too much, and also this strategy can work against highly insecticide-resistant mosquitoes. And models show and predict that this could be successful, or at least could contribute to a reduced malaria prevalence in field settings. It's important, and that's what we're trying to do at the moment, in order to avoid parasites resistance in mosquito stages, to use combinations of drugs like is done in humans. And so that's one thing that we're trying at the moment, is to look and screen for additional antimalarials, additional compounds that can kill the parasite during mosquito stages that could be used in combination. And also one thing that is important to note is that something like this could also work against outdoor biting mosquito populations, because in principle we could place antimalarials on multiple surfaces. It doesn't have to be confined to mosquito nets. And moreover, this strategy could also help or contribute, at least in part, to delay the spread of resistance to current human therapeutics in the parasite, because if you can imagine of attacking the parasite from multiple angles, like attacking the parasite during mosquito stages with antimalarials on nets, and then attacking them with drugs in humans, then that would make the life of the parasite pretty difficult. And so developing resistance to multiple drugs in multiple stages might prove complicated, even for a parasite that is so skilled as Plasmodium falciparum. Finally, I want to thank the people that did the work, in particular Doug, Christine, Rob, Maurice, and Perrine, and also all our collaborators, and surely the sponsors that are generously helping these studies, the Bill and Melinda Gates Foundation, JHMRI, and the NIH. And thank you for your time.
Okay, we have time for a couple of questions. Already over here.
Thanks for a lot of amazing talk. My name is Song Jae-cha from Johns Hopkins Malaria Research Institute. I have a couple of questions that maybe the other may have a similar kind of questions. One thing is, how much is stable of the tobacco-treated net? Doesn't have any repelling activity of the mosquito landing. The second one is atovaquone is kind of the big the drug for the treatment of the malaria. But do you have any the plan to inhibit the selection of the resistance against the atovaquone in the mosquito parasite?
So I only understood the second question.
Yeah.
The first question was, is there any effect of atovaquone on the mosquito landing? Is there a repellent effect?
I
Yeah.
see. Okay. So we, from what we can tell, we haven't done those studies, I should say. So we haven't done any, we haven't looked at the behavior, whether the mosquito behavior is modified by exposure to atovaquone, whether there is any repellent effect. But we cannot, from what we can tell, in our assays, there is nothing. Mosquitoes are pretty happy to sit on the surface. So we don't see any obvious effect, but that's something that we will need to do, probably maybe not with atovaquone or even with atovaquone itself, but with any compounds that would need to be used in the field. So that's obviously an important question. And the second question
was resistance. What are you doing to prevent resistance?
So yeah, so the idea is obviously parasites, they've developed resistance to every drug that we used against them. And so a way to maybe not prevent, but slow down resistance would be to use combination of compounds. And this is actually a very interesting question for us to understand what's the relative likelihood of parasites developing resistance during mosquito stages versus human stages. And it's something, because mosquito stages, parasites are much more constricted in terms of numbers. There are many fewer parasites in a mosquito than in a human. So we would like to model how this could emerge. But definitely for prevention, using different drugs, using combination of drugs, and different drugs than using humans. That's very important.
Yeah, Lou Miller, NIH. I'm very happy to see your work because there's such a history in Italy of great scientists who have made so many contributions over 100 years. And although Mario has died, there are others coming along. I'm very happy about that. That's not my question. But that,
Thank you.
I
Thanks, Lou.
There are others waiting, so go ahead.
Yeah. So one thing that worries me is the obvious thing, because atovaquone develops resistance very quickly. I know it's the blood stage, but when you talk about a lot of mosquitoes, a lot of parasites over time, it's a concern. Now, they combine it with daraprim, and that was worked out by a friend of mine, and it cuts down on resistance. It doesn't stop it. And it, of course, increases the cost and everything. So I really worry about it. That's my main concern, is parasite resistance over time. You know, there was a repository of a anti-malarial that was injected, and the first year was magnificent. Malaria almost disappeared. Next year, all the parasites were resistant.
Yeah, it's a major issue, right?
Yes, I definitely agree.
Yeah.
I agree with you. So it's important to test different compounds that can kill, that could be used in these strategies, definitely, and use drugs that are different mode of actions that are not currently in use in humans. So there's the need to screen for that. Yeah.
So, the questions here will go back and forth. If you could keep the questions relatively short, I'd appreciate it.
I'm sorry.
Okay. Well, you are permitted to have a long.
Okay, I'm trying to keep my questions very short. So I'm Han-Jun. I work at mitochondrial function in malaria parasites at Drexel. So I have two questions. So do you think, like, if patient take atovaquone or other atovaquone combination drugs, and the mosquito bite that patient, the amount of drug is present in the blood meal, is it enough to kill the parasite in the midgut? And the second thing is that when you see the oocysts become bigger, do you know, is the number become bigger, or is the individual sporoids become bigger?
Yeah, so very quickly. So there are studies that have shown that ingestion of autocon can block parasite development. I don't know whether the concentrations that are taken up by a human that would be effective. What we see in our hands is that the parasite is killed at the O-kinase stage, so not immediately. So actually, we should try those experiments. We don't know enough, but probably. I mean, by just feeding autocon to mosquitoes, we can see an effect. Others have seen an effect. I don't know. I don't know in terms of people taking the drug. And the second question, in terms of size of the parasite and number of parasites that are developing, we see that the number of parasites is the same in control groups. So it's just that those parasites are developing at a faster rate, but the final number of sporozoites that they develop per oocyst is actually the same. In our calculations, we think it's not that they develop more. Yeah.
Yeah. Hi, very nice talk. Prakash Srinivasan from Hopkins. You said at the outset that the insecticides, the mode of action is through binding to the legs, and it gets absorbed because they are hydrophobic. How do you think atovaquone gets in the mosquito? That's question one. And the second is, have you looked at what stages are affected that could tell you whether it's a direct effect on the parasite or on the mosquito, even though you're not observing it at the level of fecundity? Maybe it's something else is being affected that indirectly affects the parasite.
Yeah, so in terms of how the insecticides are taken up, so I actually, I don't know that much it's about it, and I've also asked a lot of people that work on insecticides, and because the cuticle of the legs is a waxy, so the legs is like covered by a waxy layer that in a way that is hydrophobic, and that's the purpose of the mosquito cuticle. And so lipophilic compounds have more chances to be taken up by the female, and that's how also drugs could be taken up. That's all I can say about that. And for the other question, which
the effect of the drugs on the mosquitoes.
So yes, we don't think that actually the drug is doing anything to the mosquito because we do see that the drug is also effective if we give it after the mosquito is already fed, and both before and after it targets the parasite exactly at the same stage, which is the sort of the zygote to O-kinase transition, and so we don't think there is something happening at the level of the midgut, that the modifications of metabolism in the mosquito that are inducing this effect. Also, we have some evidence that the mitochondria is directly targeted in the mosquito, which suggests that it's actually a direct effect.
Okay, so I have two more questions.
Yeah. Miho from Water Read. Do you think that atovaquone treatment also affects to the pre-existed oocyst or sporozoid? Because what happened in the field is like that things also.
Yeah, so that's a very good question, because all of the studies that we do target first target the initial stages of infection, so target the establishment of infection. But what about mosquitoes that are already infected, already have oocysts or even sporozoites? And those are more difficult experiments to do, especially logistically, because we need to work with already infected and potentially infectious mosquitoes. So it's taken us quite a lot of time to get the right permits in Harvard to do those experiments. But now we have those permits, and we started looking into this question. But I don't have results yet. But it looks like I don't have results to show. But it looks like the parasite is somehow affected. But I cannot really answer this question fully right now.
Okay, last question.
Kristen Lane, NIH. Most of the ladies from the right took my questions. However, from the 12% on the last data slide that you showed that had oocysts, did you examine any salivary glands to see if they had sporozoites? And second, this is a fantastic proof of concept experiment and set of experiments. It's lovely.
Thank you so much. Yeah, so we were a little bit disappointed when we saw those like single oocysts in those guts, but we didn't have the opportunity to follow them through sporozoites. Also, doing those experiments is quite tricky because when we take mosquitoes from the field and we put them in the lab, they don't like to feed. At least not in our hands. I know that Carolina Barillas has had much more success, but for us we had to feed more than 1,000 mosquitoes to get like 100 to really take the blood. So but we would like to do that. But a way that we are trying to address that problem is to take those. We actually have those parasites now back in Boston, and we are culturing them. And we would like to subclone them because also I didn't mention, but those are polyclonal infections. And also that year, last year, there was a lot of Plasmodium malariae in the region near Bobo Dioulasso, and so we don't know whether those are falciparum survivors or whether are malaria parasites or whether it's those oocysts were due to insecticide resistance or to some genetic factors in the parasite. So we would like to address this question and also test whether those oocysts are infectious. I mean, sporozoites from those oocysts will be infectious.